Students, good evening. Can you hear me? Hello, my dear all. Can you hear my voice? Am I audible? Right. Shall we start? Right, all of you, welcome to Brilliant Qatar. Now, in this academic year, we have planned to discuss MCQ question. It is a 16 days challenge, MCQ challenge. As you all know, in our final examination, we have 16 MCQs, right? For 12 MCQs and 4 accession reasons are there. Now, in this video session, molecular base of inheritance, most important chapter MCQs question we are going to solve. So all of you students, are you ready? Shall we discuss? And moreover, remember all of you, most important and H1 questions are selected, my dear kids. If you know the answer, please comment. Okay, can you send your what they call answer. Now, so I'm going to start with the first question. Look at the first question. First question they're asking. Look at the first question. RNA molecule and anticodon pairing is given. Anticodon pairing. RNA molecule, that means RNA molecule. Which RNA? It is a mRNA molecule. Here I am writing mRNA molecule. mRNA have, tell me, codons, right? Codon. Now here anticodon is given here. The second column anticodon is given. Which of the following tRNA molecule does not exist? They are asking, student. Does not exist. Can any one of you tell me how many total codons? How many total codons? Tell me. 64 codons are there. Right, out of which 61 codons are code for amino acid. How many amino acid? 20 amino acid, right? So out of which 61 codons are code for what? All 20 amino acid. 20 amino acid. What about rest of 3? We know the rest of 3 are called stop codons. What are stop codons? Tell me. UAA, UGA, UAG are the stop codon. So this stop codon does not code for any amino acid. You got my point? This anticodon, this codons, whatever these 3 codons are there, does not code for any amino acid. So they are asking which anticodon of tRNA does not exist. Can any one of you answer this one? Let's take. Here it is. UCA, AUG, UAA is given. UAA. What is the anticodon of this one? This one I can write UAA. UAA is a stop codon. Now codon is this one. AUC is the codon. What is the anticodon of this one? UAG. UAG, UAA are stop codons. So tell me which of the following 4 does not exist. Tell me. Now answer is 3 and 4. Where is the 3 and 4? Answer option C. This 3, that means this 2 amino acid anticodon does not exist. So why they does not exist? Because they does not code for any amino acid. These codons are not code for any amino acid. You got my point? So what is the correct answer? Correct answer is 3 and 4 R does not exist. Remember student, in molecular base of inheritance, this type of question you should practice. How many total codons? 64 total codons, out of which 61 codons code for all 20 amino acid. 3 codons are stop codons. As we know that called stop codons, right? Now the first question answer is option C, 3 and 4. May I know how many of you given answer? Are you there? Come on, answer me. Are you there, students? Right. Look at the next question. Next question. Which of the following statement about untranslated region we commonly call UTRS? Untranslated regions is or are true? They are asking. Untranslated regions. Tell me, untranslated regions are seen in where? mRNA. Yes or no, student? Untranslated regions are seen in where? mRNA, right? Now look at here. Where is this untranslated region? Before start codon, after stop codon, correct? Then look at the first one. Present on rRNA. Is this rRNA or mRNA? Is this rRNA or mRNA? Tell me. It is mRNA, right? So this is not correct. Next, present on mRNA 3 position only. Wait. All of careful, look at here. This is mRNA. Okay, this is mRNA. We know 5 prime to 3 prime, right? 5 prime to 3 prime. At 5 prime, we know there is a cap is there. This cap is made up of what? Tell me. Methylated guanosine triphosphate. And here it is tail is there, beta. You know, this is called tail. Tail made up of polyadenine. AAA. How many? 200 to 300 many, what they call adenine. Adenylic tails are added, right? It's called capping and tailing. Now, this is called AUG, start codon. here UAA stop codon. Before start codon, after stop codon. That means fifth place also there, third end also there. But what is the question better here? Second one, present on mRNA at third position only. No. Next, present on mRNA at fifth position only. No. What about the fourth statement, student? Fourth statement is present on mRNA at both stages. Correct or not? Yes, sir. This statement correct. The statement correct. Next, not required for the translation process. UTRS important for what? Increase the efficiency of translation. Yes or no? So this is also not correct. What about the last one? Required for the efficiency of translation. Come on, answer me. What is the right answer of this question? Come on, Syed Ahmad, Pooja. What is the right answer of this question? Aditya, what is the right answer of this question? The fourth and sixth statements are correct. So I am going with option D. Am I correct? Am I correct? Yes, very good. Shabas. Very good. Shabas. Next question. Question number 3. This is, they're asking UTRS. Remember, student, UTRS increase the efficiency of translation. 2 UTRS are there at 5th prime and also 3rd prime. And remember, cap is made up of unusual nucleotide. It is called what? Methylated guanosine triphosphate at 5 end. Tail made up of polyadenine at 3 end. Remember, that's called tailing and it is capping. You got my point? Question number 3. I hope you all are giving us right. Nayan, correct. Right. Pooja, done. Look at the next question. Third question. The regulation of gene expression. Gene expression, we know what is the meaning of gene expression? DNA to mRNA, mRNA to protein. That is called gene expression, right? The regulation of gene expression in eukaryotes can be extended at. It's asking eukaryotes. We know in prokaryote gene regulation can be regulated at only transcription level, right? Transcription level, we know, we have seen in the lac operon process at exactly mRNA synthesis place that what operator gene, no, operator off. You got my, because of that one RNA polymerase enzyme could not slide over ZYA genes, right? Structural genes. But they're asking eukaryotes. Can any one of you got the answer? Answer me. Answer me. What is the right answer of this question? In case of eukaryotes, gene expression at transcriptional level, correct. Processing level, correct. Transport of mRNA, correct. Translational level. In case of eukaryotes, I told you this one in the theory class, we discussed many times. In the eukaryote, the gene expression could maybe any one of this level. Not only a particular transcription level, not only particular the processing. You know what is the meaning of this processing level? You know what is the meaning of this processing level? Splicing. Splicing is the process we know from hnRNA to mRNA formation. Nonsense. That waste part of introns are deleting, exons are joining. So answer me. What is the right answer of this question? All of these answer. D is the right answer. Remember, my dear kids, in the exam if question come, in case of prokaryote, the gene expression regulated at transcriptional level. Remember, in case of us or in case of eukaryote, it could maybe any level. It stop. Don't express now. This is not right time to express. So all gene, particular gene will be not going to express. This is called gene regulation mechanism. Lac operon, especially lac operon, this academic year lac operon, very high chances there. So all of you practice lac operon question also. Next question. Next question. Look at here. This is also on a small question from transcription level. Can any one of you? For initiation, the ribosomes bind to A, don't know A, at the start codon and B is recognized by C. Choose the correct option for A, B, C is given. Ribosome attached to where? Come on, all of you. Who is this? Chris. What is the ribosome? Where is ribosome attached to? Come on, all of you. Pooja, answer me. Where is this ribosome? mRNA. Yes or no? It's attached to what? All ribosomes attached to mRNA. But mRNA in option A also there. Option B also there. What is? So option C and D we can eliminate. Option C and D, it's not answer because ribosomes are attached to mRNA, messenger RNA, right? Now what is this? Option A or B? Which one is correct? Next, here at A, start codon. Almost we got the answer. Start codon. Where is the start codon? Tell me. Yes, where is the start codon? AUG. So second one is given. Second one also AUG is given here. Next, C is given here, initiator RNA. Here only simple RNA. Initiator RNA and terminator RNA. Two RNAs are there, tRNAs. Initiator RNA can initiate the translation process, and terminator factors, terminator RNA. That is what they call terminate what? Translation process. So what is the correct answer? The right answer is option A. Remember all of you, correct answer is option A. What is polysome, student? In prokaryote, many ribosomes are combined, attached to whom? One single mRNA, polysome formation. Here also, we have a smaller subunit and larger subunit RNAs are there, right? RNA, ribosomes are there. These ribosomes are combined, attached to what? mRNA. So, start codon, AUG. Next, initiator, tRNA. Fine. So, right answer is option A is the correct answer. Am I clear till here? Everything clear? Pooja, done. Next, right, Ansh, next 1 minute. Next question. Look at the next question. How many of you give correct answer of this question? Look at it. Carefully read. Carefully read what is given. Name the enzyme that facilitate opening of DNA helix during transcription. Which enzyme will opening this DNA helix during transcription? So I think you people can guessing this is the helicase enzyme, right? So what is the right answer? Come on, answer. Try all of you. What is the correct answer? Come on. DNA polymerase. DNA. My dear, DNA polymerase enzyme in DNA replication, beta. They are asking what? Transcription. They are asking transcription. They are not asking DNA replication. Are they? No, right? Now read. Carefully read. There are DNA helix during transcription. Ligase enzyme. We know ligase enzyme function is to join the two DNA particles. Done. Now DNA helicase enzyme. DNA helicase enzyme can unwind the DNA ORI site at ORI site in where? DNA replication, right? But they are asking transcription, my dear. Any one of you? Correct answer? Yes. The answer is what? RNA polymerase. DNA polymerase and RNA. Answer is RNA polymerase. Don't get confused, student. They did not ask about the replication. They asked about the transcription. We know this RNA polymerase enzyme go and bind to particular area. What is that? Promoter. Do you remember this? Promoter and this open unwinding. They did not ask about the replication. They asked about the transcription. So our answer is what? RNA polymerase enzyme. One more important thing keep in mind. RNA polymerase enzymes are, tell me how many are there in case of eukaryote? RNA polymerase enzyme. RNA pol 1. Next RNA polymerase 2. Next RNA polymerase 3. RNA polymerase 3. Even in DNA polymerase also, DNA polymerase 1, 2, 3 are there. Here RNA polymerase 3. Tell me, RNA polymerase 1 is essential for the transcription of rRNA, ribosomal RNA. RNA polymerase 2 is hnRNA or, tell me, mRNA. Yes or no, student? mRNA, messenger RNA. RNA polymerase 3, tRNA. Now answer me, all of you. In the exam, if question come, which of the following RNA polymerase enzyme can transcribe all these three type of RNAs in bacteria? One only, RNA, simple RNA polymerase only. If they ask in eukaryotes only, you should be keep in mind 1, 2, 3 different type of RNA polymerase enzymes are there. Are you with me? You got my point. Now, so the answer is what? Option D is there. Most important thing, RNA polymerase enzyme during the transcription initiation process, they go and attach to where? Promoter. Remember all of you, they attach to promoter. Easily remember PP, my dear. Polymerase and also promoter. Got my point? Shall we go to the next question? Next question. What about this question? This question, they are asking few steps involved in polypeptide synthesis. That means protein synthesis. Polypeptide protein. Carefully read this question, okay? Polypeptide synthesis are given below, okay? In which of the following step does tRNA participate is given. tRNA participate. In which all these steps tRNA participate? They're asking what? Polypeptide synthesis, protein synthesis. Chalo, we'll see options. Activation of amino acid by binding with amino acyl tRNA synthesis. Can you please tell me in step number 1 tRNA required or not? Say answer yes or no. tRNA required or not for the first process? Come on. Any one of you? For the first process, yes sir. Answer is yes or no. tRNA is requirement. Yes, very good. It's correct. Amino acylation by binding of amino, what they call activation of tRNA. It is correct. tRNA requirement is there. tRNA will involve. What about the option 2? What about the option 2? Elongation of polypeptide. Elongation of polypeptide means what? First 3 codon. First codon is over. That means AUG, start codon. So start codon particular tRNA bring that amino acid. Next different tRNA will bring the next amino acid. Next. So this is called elongation process. For this elongation process also we need tRNA. Yes or no? Then who will bring the amino acids? If tRNA is not required, then who will bring the amino acid? Second step also tRNA required. Next, translation of mRNA to form polypeptide. Yes, it is important. The process called translation. This process is known as what? Tell me. Translation. For this process tRNA required. But question number 4, option 4, transcription of DNA to rRNA. DNA to RNA. Transcription from DNA to RNA. In this process, tRNA required, student? Come on, answer me. In this process, tRNA requirement is there? Come on. Come on, answer me. In this step, fourth step, tRNA requirement is there. Sir, no, this process takes place in the nucleus. tRNAs are in the cytoplasm. Protein synthesis process takes place in the cytoplasm because ribosomes are outside. All free amino acids are outside. So, tRNA outside. So tell me what is the right answer of this one. In this process we don't want tRNA. So what is the correct answer? Tell me. 1, 2 and 3. Where is the 1, 2, 3? 1, 2 and 3. Option D. Am I correct? Till here everything clear? Are you with me, students? Done? Right. Good. Very good. What about the next question? Look at the next question. This question. UTRS, untranslated region, present on mRNA refers to. Very simple. We already done this one. Think all of you and find out the answer. Correct answer. Come on. Atul, Pooja, Nayan, I need answer. Try this one. What is given here? UTRS, untranslated region present on mRNA refers to what? Untranscribed region at both 5 end and 3rd end. Untranslated region 5 end, untranslated region at both 5 end and 3 end, untranslated region at 3 end. What will be the right answer? What will? Very good, absolutely correct. Very good, student. Option C is the right. So look at the first one. First option. What is the first option? Untranscribed region is given. What is this untranscribed region? Untranslated region, right? It is because it does not undergoing translation process. So answer is C, not A. Don't get confused, my dear student. Answer is C. I am clear? Right, almost all. Very good, Sabas. Absolutely correct. Very good. Next question. Look at the next question. Now what about this one? Simple question. Very simple question. I hope all my students can give this answer. Which of the following rRNAs act as structural RNA as well as ribozyme in bacteria? My dear kids, we learn in the 11th grade. We learn in the molecular base of inheritance initial classes. Ribosomes made up of 2 things only. Ribosomes made up of 2 things. Can any one of you guess what are these 2 things ribosomes made up of? Ribosomes made up of rRNA and proteins. Now ribosomes made up of rRNA and 80 different type of what? Protein. This 80 different type of proteins are inactive first. Now here this rRNA is behave like a enzyme called ribozyme. It is a catalyst, biocatalyst. If you are able to recall biomolecule chapter 11th grade, all enzymes are tertiary protein except ribozyme. Ribozyme is not a protein, it is RNA. It is RNA. So we can write, what tell me? Ribozyme, 5s rRNA, 18s RNA, 23s rRNA, 5.8s RNA. Any one of you? Any one of you? Absolutely correct. Who is this, Pooja? Correct. Next, Chris, what is the correct answer of this one? Which is going to behave like a ribozyme? You know the function of ribosome, kids? Ribozyme is the enzyme essential for dash. Any one of you? This ribozyme enzyme essential for something. My dear, this ribozyme enzyme is the one which is going to make peptide bond in large subunit of ribosome. We know peptide bond formation and polypeptide chain is going to form in the largest subunit of ribosome, right? Then who will make a peptide bond between two amino acids? Ribozyme enzyme is essential for that. Remember, all of you. So answer is 23s rRNA. How many of you given correct answer? Yes, 23. Atul, correct. 23s rRNA is behave like a ribozyme. It is an enzyme. This enzyme is actually it is RNA, but work like what? Behave like a enzyme, biocatalyst for the formation of peptide bond in ribosome. Which ribosome? Large subunit of ribosome, right? So answer is option C. What about this question? What about this question? Select the incorrect statement they are asking. Select the incorrect statement from the following. Incorrect statement they are asking. Incorrect statement from the following. Any one of you, try incorrect statement. What is given here? In human genome contain 3164.7 million nucleotide bases. That means 3.1 billion of bases, right? Next, less than 10% of the genome codes for protein. Next one is given, repeated sequence make a very large portion of DNA. Chromosome number 1, most gene 2968. Y chromosome holandic genes are 231. What is the incorrect? Something is incorrect here. Can any one of you in this question? One statement is incorrect. If you know proper about the HGP, Human Genome Project, come on fast. Yes, good. In Human Genome Project clearly mentioned what? Less than 2% of DNA. But what is given here? Less than 10% of genome code for protein. No, less than 2% only. Less than 2% only, right? So what is the right answer here? The incorrect statement is option B is the incorrect statement. Are you with me? Correct. Which is the largest gene? Dystrophin. Largest gene located on chromosome number 1. Largest gene, dystrophin protein synthesis. That gene located on chromosome number 1. Can any one of you tell me how many nucleotide? 2.4 million bases, right? Next question. What about this question? What about this question? You need to think this question. Students, carefully read and find out the answer. Carefully read and find out the answer. Come on, answer this one. Try to answer this. If there are 999 bases in an RNA that code for protein with 333 amino acid. 333 amino acid, right? Amino acid. How many bases are there? 999 bases are there. Nitrogen bases. We know 3 base become 1 codon. 1 codon equal to 1 amino acid. So 999, 333 amino acid. Correct. Okay, fine. And the base at position 901 position deleted such that the length of the RNA become, now 901 nucleotide is what got deleted. Now this 999 become 998. How many codons will be altered? They're asking. 1 to 900, no problem, kids. 1 to 900 bases, no problem. 901 deleted. From there to 999, that means 998, problem is there. That means how many amino acids will be get altered? Fast. How many amino acids get altered? Try. Think, Pooja, think. Reem, think. Chris, try to answer. Think. How many total codons are getting altered? How many total bases are getting altered? How many amino acids finally? Come on. Dystrophin, okay, fine. Answer. Look at here. 1 to 900, no problem. 1 to 900 bases are good. That means, shall I call 300 amino acid? So first, out of 333 amino acid, 300 amino acid, no problem. 300 amino acid polypeptide chain will be synthesis. No problem. Where is the problem? 901 problem. So that deleted. From there to 998 got affected. That mean 33 code. Yes, absolutely correct. 33. Are you with me? You got my point. This is called a deletion type of mutation. Where exactly mutation? 901 place mutation. Till that place everything fine. That means 300 amino acids will synthesis. From 901 got deleted. From there everything will be changed. That means how many? Rest of all. That means 99 amino codons will be changed. That means 99 bases are changed. That means 33 amino acid. You got my point? Yes. Answer is what? Tell me. Option B is the right answer. How many of you got this? How many of you? 33. Yes, Pooja. Correct. What about this question, students? What about this question? Removal of. Very easy. Everyone should answer this one. Removal of RNA polymerase 3. I told you, RNA polymerase 3 function, I told you. What happens if you remove this? RNA polymerase 3, if you remove from nucleoplasm, will affect the synthesis of what? Will affect the synthesis of which RNA is not going to be synthesis? Come on. Which RNA not going to be synthesis? Fast. Here, tRNA, hnRNA, mRNA, rRNA. 4 options are given. I told you rRNA, which enzyme? RNA polymerase 1. hnRNA and mRNA both are RNA polymerase 2. What about this one? Smaller. This one is called RNA polymerase 3. tRNA, also called smallest RNA. Dissolved RNA, adapter RNA, clover leaf-like structure RNA. For the synthesis of, for the transcription of tRNA, we need a specific enzyme called RNA polymerase 3. They're asking what happens if you remove that. If you remove that, there is no production, or there is no synthesis of tRNA. If tRNA is not there, who will bring the amino acid? So automatically protein synthesis process will be get effect. Are you with me? You got my point? Next question. What is the correct answer of this one? tRNA. Answer is option A. What about this question? Come on. Can you answer this question? Choose the incorrect option from tRNA molecule. They are asking incorrect statement. Rest 3 is correct. Incorrect statement about the tRNA molecule. Find out. It has an anticodon loop that has base complementary to the code. It has an amino acid acceptor end which is bind to amino acid. tRNA are not specific for each amino acid. tRNA look like clover leaf-like structure. What is the correct answer? What is the right answer of this one? Any one of you? Fast. Then look at the option C. What option C is given? Let's take one small mRNA that we can get understand. AUG, UUU, UUC, UGG. I'm taking UAA. Stop codon, right? So for this codon, but particular tRNA will be there, and this particular tRNA. Now answer me with anticodon. What's the anticodon here? UAC. Here it is A, here it is G, here it is C. Now every tRNA have a specific anticodon. That specific tRNA will come, read, okay, I understand this is my anticodon, I understand I need to bring phenylalanine, I'll go and bring the phenylalanine, I need to bring the tryptophan, I need to bring that methionine, so on, Sami. Every codon have a specific anticodon tRNA in the cytoplasm except 3 codons. Except stop codon. Stop codon, there is no tRNA. Stop codon will come at the end. Ribosomes will wait. Any tRNA can bring the particular amino acid. No RNA, no tRNA is responding because anticodon is not there. So I am not specific to this codon UAA. I am not specific to this codon UGA. So there is no amino acid bringing process. So automatically it will, what they call, translation will be terminate. So what is the right answer? Answer is option C. tRNA not the specific. Each tRNA for each codon they are specific. You got my point? Yes. Hi. Who is this? Right, Lisna ma'am and Pooja ma'am. All of you. Hello. All of you. Next question. What about this? Under which of the following conditions will there be no change in the reading frame of following mRNA? Which condition mRNA is not going to change? Carefully read this question. Student, this type of HOT question, statement based question, incorrect, correct question, you should prepare, you should perfect before the final exam. Read the options carefully one by one. What's the first question? What is given with here? AAA, AAC, AGC, GGU, GCU, AU, U. Fine. So first, let's take here deletion of G from 5th position. What is the 5th position? 1, 2, 3. This is 3C, 4, 5. 5th nucleotide G we are deleting. So if we delete like that, if you delete, answer me, AAC become 1 codon, ACG become 1 codon, GUG become 1 codon, CUA become 1 codon. Look at UU. What is this? They're altering. They're getting problem. They're getting change. They're changing, right? Do you remember frame shift mutation, student? Insertion of any one nucleotide or deletion make change in the frame of mRNA. So option 1 is not correct. They are asking which one is no change they are asking. So this is not correct. Come to the option B. Insertion of A and G at 4th, 5th position. Okay. A 1, 2, 3, 4. 4th place, one more A, and also 5th place we are adding what, tell me, G. This condition also changing because 2 nucleotide only you added. You got my point? 2 nucleotide only you added. So it will change. So it is not correct. It is not correct. Come to the third. Deletion of GGU from the 7th, 8th, 9th. So this one, GGU, where is GGU? This one entirely I am deleting. This will become 1 codon. This is 1 codon. This is 1 codon. This is 1 codon. There is no change. Yes, correct. Yes, students. You got my point. So there is no change. So this appropriate answer. What about the last? Insertion of G. Whenever you insert one nucleotide, my dear kids, it will change the frame of mRNA. When you delete one also, that's why this is clearly given the evidence that codons are triplet. This is clear, giving the evidence codons are triplet made up of 3 nucleotides. Remember, so answer is what? Option C is the right answer. What about this? What about this? Look at here, most important question. Diagrammatic questions can come, student. This diagrammatic questions from molecular basis. You should practice this all question. Can any one of you tell me which process is this? Which process is this? Eukaryotic, what given with the transpiration process in eukaryotes. Sometimes if the question did not given it is a prokaryotes or eukaryotes, just given a transcription, how can we identify it is a eukaryote? I told you splicing is there, capping is there, tailing is there. Look at their capping, tailing, everything is there. If it is there, it is what? Eukaryote. You got my point? Now, okay, what is given here? Question. We should identify A, B, C, D. They are given A, B, C. What is this A enzyme? This area I call promoter. To promoter A is attaching dark shaded B area, light area C, and where is D? This is called D is given. What is the correct answer? What is the correct answer? Any one of you? Any one of you? Come on, Reem, try this one. First, I can tell beta A is RNA polymerase because RNA polymerase enzyme only can what? RNA polymerase enzyme only can transcribe. So A is RNA polymerase enzyme. B, C. Which one is intron? Which one is exon? How can I identify? My dear kids, darkly shaded one is always non-functional. That means we can say what? Tell me, intron. Darkly shaded part is always intron. It's not going to make a protein. We should take that out. Should take off. We don't need that one. So answer me. B is I'm writing intron. C is light one is called exon. Exon is useful or it's going to make a protein. Splicing. Now tell me here what happened. Here D. D is what? Tell me. Polymerase. A tail. What is the right answer? What is the right answer of this question? Come on. Right answer is what? Now, RNA polymerase 2, exon. It is exon is given. Where is B? B is not exon. Leave it. This question option leave it. A, RNA polymerase 3, RNA polymerase 2, right? It's RNA polymerase 2, right? Now I'm writing RNA polymerase 2, intron, exon, polyadenine tail. Option C. Am I correct? How many of you given C answer? No. Reem, look at here. How it is become B? There RNA polymerase 3 is given beta. RNA polymerase 3 is not for mRNA production. mRNA synthesis, RNA polymerase 2, right? It is not RNA polymerase 3. They are giving here it is mRNA production. That means this must be RNA polymerase 2 beta. So answer is what? The correct answer is option C comes. Clear? You got my point? Yes. Good, very good. Next, what about this question? This type of diagrammatic question should not get confused in the examination, student. That's why before examination this 16 days MCQ challenge to discuss different type of questions to rectify our mistakes. Where I am doing mistake, oh my god, this is the particular area I have done mistake. So one mistake cannot bring you 100 out of 100. You understand my point? What about this question? Any one of you? In bacteria, transcription and translation takes place in a same compartment. Why? Why it is in same compartment? In case of eukaryotes, what happened? Transcription exclusively in the nucleus, translation in the cytoplasm. We know the reason because nuclear membrane is in everything. In the bacteria, that nuclear membrane, the prokaryotes. Naked DNA is there, no nucleus proper, no nuclear membrane is there, no membrane bound organelles are there. Yes or no, students? Read the question. Options: no separation of cytosol and nucleus, correct. mRNA does not require any process. What is the meaning of this processing? Any one of you help me out? What is the meaning of this processing? What is the meaning of this processing? Can you tell me what is the meaning of this processing, my dear kids? Okay, correct answer. Okay, fine. What is the meaning of this processing? My dear, the process, very good, absolutely correct. Splicing. So in the bacteria, no splicing, no intron removal, no exon joining, no tailing, no capping, nothing is there. So what is the right answer? This is also undercome. Yes. And due to the presence of nucleus, where is the nucleus in prokaryotes? No, the answer is option C. Moreover, my dear kids, keep in mind, open the NCERT, clearly mention that mRNA will be started to undergoing translation process before it fully matured in case of prokaryote. Before it fully matured. But in case of eukaryote, mRNA has first hnRNA has to form. hnRNA splicing mRNA. It has to come through nuclear pores from nucleus to cytoplasm, ribosome and start. So the right answer is option C. Very good. What about this? What about this? Very easy. Very easy. What happens in the tailing process of transcription? what happened in the tailing process of transcription? Any one of you? Anyone? Chris, try this one. Reem, try this one. Adenylate residue added at 5 end of RNA. Sir, 5 end is not tail, right? 5 end is what? 5 end is a cap. It is a cap. So this is not correct answer. Adenylate residue added third end of RNA. Guanylate is no, leave it. Guanylate, no, leave it. CD is not correct. So it must be, yes, absolutely correct. What is the right answer, Atul? Right answer is what option? Yes, option B. How many adenylate this one? 200 to 300. Polyadenine tail. Polyadenine tail. So the right answer is option B. Are you with me, students? Yes. Good. Very good. What about this? A very easy question. Everyone know this. I hope, I believe. In bacteria, which enzyme catalyzes the transcription of all type of RNA? mRNA, I told you this one. tRNA, rRNA. Come on, answer. Which one is going to synthesize all different type of RNAs? Come on, answer. Option is given. DNA dependent RNA polymerase, DNA dependent DNA polymerase, RNA dependent RNA polymerase, RNA dependent DNA polymerase. Come on. Answer is C or B. C or B. It must be B. DNA dependent. So DNA dependent RNA polymerase enzyme, right? It must be A, right? Is it A? How come it's C, my dear? It is A, right? Look at here. It depends on DNA to make RNA. DNA dependent RNA polymerase enzyme. DNA dependent RNA polymerase enzyme. So answer is what? Option A. But I have a question. What is this RNA dependent DNA polymerase? Any one of you answer me. This enzyme is present in which type of viruses, students? If you give answer, sure you will get 99 in final exam. If you give answer of this question, that means you properly revise the biology. RNA dependent DNA polymerase enzyme means what? Yes, absolutely. Who is this? Absolutely, very good. It is actually reverse transcriptase enzyme from RNA to DNA formation. HIV. You got my point? Good, very good. Absolutely correct. Very good. Sabas. What about this? Name the nucleotide added. Fifth end of HNRNA. Fifth end of HNRNA, some unusual nucleotide added. What is this unusual nucleotide? What is this unusual nucleotide? Fast word. It is what? Methylated guanosine triphosphate. Yes or no? What is this methylated? Ethyl, no, ethyl, methyl guanosine triphosphate, MGTP. How simple. It's a cap. Good, very good. What about this question? Which of the following RNA should be most abundant in animal cell? This type of questions can come, student. You should be perfect. Which one is smaller? Which one? Write down all of you. First you should remember rRNA is the abundant RNA. tRNA is the smaller RNA. mRNA is what? Tell me. Short-lived RNA. Remember, short-lived RNA. This tRNA we can also call snRNA or siRNA. You got my point? Remember. So what is the abundant one here? Abundant RNA is what? rRNA, ribosomal RNA, essential for the synthesis of polypeptide in large subunit. What about this? Can any one of you simple match the following? Very easy. Choose the incorrect one. I'm sorry, it's not a match the following. Incorrect pair they are asking. Incorrect. Something is incorrect here. Promoter binding site for RNA polymerase. I told you how to remember. PP, promoter polymerase. So this is correct. Let me check. My students are giving our answer. What is the right answer of this question? Find out, students. Something is incorrect here. Out of 4, something is incorrect. See, Nayan, it is C beta. Let me check. Let me check. Terminator. Define the end of transcription process. End of transcription is called terminator. Promoter, terminator. We know this. Okay. Cistron. Segment of RNA coding for polypeptide or DNA coding, my dear? RNA or cistron, sir, it is a small part of DNA which code for what? mRNA protein, polypeptide. Yes, absolutely correct. So this is not what they call, it is incorrect. That means correct answer is this. Regulatory gene do not code for any RNA or protein. Yes, regulatory genes will regulate. You got my point? 98% of genes are regulatory genes only. So answer is what? Tell me. Option C is the incorrect statement. Cistron. You know, monocistronic, polycistronic. That means one mRNA to one polypeptide, monocistronic. Example, eukaryote. One mRNA more than one, polycistronic. Any one of you give me the example of polycistronic kids. One example of polycistronic. Anyone? Polycistronic. Yes, good. Come on, tell me. Polycistronic example is lac-operon. Yes, lac-operon. Sabas. What about this? If both strands of DNA copying during transcription, both DNA strands. Do you remember that day I told you? Yes, absolutely. Tell me. Here, this is a DNA sequence, my dear. This is called 3', 5'. What about this one? Complementary is what? Complementary is 5', 3'. Tell me, first one is called template or non-coding strand. Non-coding. 5 to 3 is called what, beta? Non-template or coding strand. What happened if both undergo transcription? I told you out of 2, one only undergo transcription, other one will not. Tell me, I clearly mentioned the protein information encrypted in where? In coding strand. To get that coding strand in mRNA, we have to transcribe template strand. That's why template strand and mRNA almost the same. In place of T, you only change, my dear. So what happens if both undergo transcription? Chalo, we'll read. If both undergo transcription, this one undergo transcription, let's take this one make mRNA. Okay, this one also make mRNA. Both will become double-stranded dsRNA form. dsRNA never become protein. We know, never become protein. Saying RNA only become protein, but it is a double strand RNA. RNA must be what? Single strand. Chalo, we'll read. The segment of DNA would be coding 2 different protein. Absolutely correct. One will become one protein, other one will become other protein. We are looking for different protein, one protein only. We are looking for protein from this strand. What about this one? If this one also undergoing protein and making a new character, it might be, it's a difficult one. So this is correct. 2 RNA will be produced simultaneously, complementary each other. Yes. There will be formation of double strand. Absolutely correct. So answer is option D. How many of you given? D answer? Yes. Are you there? Yes, absolutely correct. What about this question? Answer this question. Carefully read and answer. Carefully read, student, and give me the answer. I'll read the question, but I'll not help you this time. Think and give me the answer. Which of the following mRNA can be transcribed? Out of the 4 options, 1 RNA only undergo transcription. Rest of 3 does not. Why? Which one? Come on. Which one? Think. Who is this? T7. Come on, answer this one. Good. Atul, why? Why A? Why not C, B and D? Think. Yes, Rim. Yes, beta. Easy? Good. Tell me answer. I'm waiting for answer. Yes. Because start code, absolutely correct, absolutely very good. Look at the rest of 3. Here UAA, stop codons are there. Start codon, methionine, UAA, UAA. UUU code for what? UUA code for what? Tell me. Any one of you? UUA code for what, beta? Leucine, lysine, valine, what? U code for what? UUA. UUA code for what? Anyone? No, beta. It is stop. This UAA is stop. What about UUA? You at least tell me UUU code for what? UUC code for what? Both are code for leucine. Good, very good. It's leucine. UUA leucine. UUC what? Phenylalanine. Good, very good. Phenylalanine. So answer option A. Very good. Sabas. Good. Can any one of you tell me GAA code for what? GAA code for what? Molecular basis, perfect if you give the answer. GAA code for what? Which amino acid? This one. GAA code for what? Anyone? Try. GAA. Sabas. Glutamic acid. Glutamate or glutamic acid. Very good. GAA both are glutamic acid. Sabas. Very good. Now, what about this question? This is Michelson-Stahl's experiment question. Simple. If you know the concept, very easy. Let me check. Here, what is given? Michelson and Stahl experiment in 1958, DNA extracted from the culture one generation after the transfer from N15 to N14 medium had a hybrid or intermediate density. Why? Semi-conservative DNA replication. Chalo, answer this one. Think. Sure, you will, my dear. Don't give up. Keep practice. 100% you will get good marks in NEET exam. Don't worry. Now, they are asking here N15, N14 medium had a hybrid intermediate because the generation time E. coli culture was about 20 minutes, because it would take 20 minutes for RNA replication, because it would take 20 minutes for the replication of DNA to RNA, because it would take 20 minutes for the translation of RNA to protein. What is the correct answer? Think, my dear. Don't do this type of mistake. It is Michelson-Stahl experiment. That means replication of DNA. In C, because of 20-minute replication of DNA to RNA. No. It is DNA to DNA. So what must be the answer? Yes. Who is this? Yes, correct. Venkata, correct beta. What is the right answer? The right answer is option A. Because generation will take 20 minutes to culture, it was one DNA to other DNA. It's 20 minutes. So answer is what? Option A. DNA to DNA formation. Got my point? Just recall better this entire concept, recall and find out your mistakes. Don't worry. Eventually you will get good marks. Now look at the next question. What about this? A molecule that can act as genetic material must fulfill the traits which below except the given. We know biomolecules are proteins, carbohydrate, DNA, RNA. DNA, RNA only considered as a genetic material. Why? Why can't protein as a genetic material? Why don't you consider as a lipid as a genetic material and protein as a genetic material and carbohydrate as a genetic material? Why are you considering DNA, RNA as a genetic material? They are having a 4 characteristic features, right? We learn. There's a 2 more question also. But they are giving one exception. What is that one exception? We'll read. Already got the answer, huh? Very good, Sabas. Let me read. It should be able to express itself, okay? It should be able to generate replica, done. It should be unstable structurally, no, it should be stable, more stable. Which one is more stable, DNA or RNA? DNA is more stable than the RNA. There are many reasons are there, but it should be stable. Now, it should be provide scope for slow change requirement of evolution. This slow change is called what? Mutation. Remember, that means except what will be unstable. Answer C. Let me check my students. Good, very good, very good, very good. What about this? What about this question? Griffith experiment. My dear kids, last year 2023 paper, there was Michelson and Stahl experiment was there. This time Harshan, Martha Chase or Griffith experiment might. So these two experiment thoroughly prepare. Last year for 5 mark, what experiment you know? Michelson Stahl experiment was there. 2023 March paper. This time, these two there is a chance. What is the correct answer of this one? Live R strain A. R is non-pathogenic, right? Rough variety is a non-pathogenic. That means here it will be survive. Yes, survive. Live S pathogenic. S is pathogenic. Streptococcus. That we can write what will be B. B is what will died. Heat killed S. Survive. S survive. Heat killed S plus live R2. That means here it is died. Am I correct? Am I correct? SD, 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 Sandeep sir, Sandeep sir. Is it correct? SD, SD means? So what is the correct answer here? We can write survive, died, died, survive. No. Survive, died, survive and died. Am I clear? Correct answer? Yes, absolutely. So I will write option B. Griffith experiments of transformation thoroughly prepare and go. You got my point? Griffith, MacLeod, McCarthy, Avery, properly prepare and go this experiment. Yes, good, very good. What about next question? Something wrong in this question. Carefully look at something wrong. They're asking what? Incorrect statement. Look at the incorrect pair. They're asking incorrect pair. What is incorrect? Find out. Think. Basic amino acid. Amino residue histone protein made up of arginine and lysine. Lysine, arginines are what type of amino acid? Basic amino acid. Right? Correct. Lysine, arginine, basic amino acid. Biomolecule chapter also we discussed basic amino acid. But that means more amino groups than carboxylic group. Unit 8 molecule in histone octamer. Correct. Negative charge DNA wrapped around positive charge histone protein nucleosome. Correct. Thread like colorless unit structure chromatin in nucleus. What is the incorrect statement? Nayan, I changed something. Read the question carefully. Read the options carefully. 720 by, yes, Reem, congratulations. 720 by 720. You are the big ambassador. Very good. God bless you with 720 marks. Don't worry. What is the right answer? What is the correct answer of this? Option D. They are given colorless chromatin. Chromo means color. Chromo means color, right? So chromatin is colorless, is given it's colored. So incorrect statement is what? Option D. Are you with me? You got my point? Yes. Next question. Oh my God, very simple. The beginners of this molecular base of inheritance also can answer. Very simple question. Any one of you? Come on, try this one. They're asking what? Diagram represent show important concept for in the genetic amplification of DNA. Fill A to see the given. DNA to DNA called DNA replication. Okay. DNA to mRNA A called transcription. mRNA to protein B called translation. Right? Translation. Listen, my dear kids. DNA to DNA formation. Enzyme name is what? DNA dependent DNA polymerase enzyme. DNA to RNA called DNA dependent RNA polymerase enzyme. Or simply RNA polymerase enzyme. And translation process is given. What is the right answer? Very good. Sabas, absolutely correct. Transcription, translation, Frederick Crick. What is that? Francis Crick. I'm sorry. So option C is the right answer. Am I clear? Very good. Very good. Look at the next question. What's something wrong in this statement, student? It is a 4 statement based question. Something wrong. Can any one of you? Can any one of you? Which of the following is not a correct salient feature of double helix structure of DNA? Something wrong is there. Line by line read. Line by line read. Who is this? T7. It is T7 or 1788. Very good. Absolutely. Very good, my dear. Two polynucleotide chain have backbone of sugar and phosphate and project, correct. Two chain have anti-parallel, correct. Adenine form 3 hydrogen bond. A to T, how many? 2 hydrogen bonds only, not 3. You got my point? G to C is 3 hydrogen bonds. Good, very good. So option C is the right answer. What about this? What about, can you answer this question? Very simple, very easy. Nucleoside and nucleotide structure they're asking. I'll be silence. Come on. Come on, come on, come on. Come on. Sir, if I get next answer correct, you will give. Yes. Come on, come on, come on, try. When a phosphate group is linked to a group of nucleoside through B bond, a corresponding C is formed. Any one of you? Any one of you? B, Rim, think. Choose the correct option A, B, C. A, 5-end OH, B, phosphodiester bond, nucleotide. Done. 3 at 3 OH, that means 3 prime OH, phosphodiester bond, nucleotide. 2, leave it this one. 5 OH, phosphodiester bond, nucleotide. This one also, leave it better. A or B, correct thing. Correct answer. Yes, edits. Correct. What is the right answer? Mac, absolutely correct. The right answer is what? It will be 5th end and phosphodiester bond make nucleus. Nucleotide means, nucleoside means what, beta? Nucleoside consists only pentose sugar. Tell me, plus nitrogen base. First carbon to the nitrogen, pentose sugar. Now here they don't have phosphate, so phosphate attached to it, 5th carbon of pentose sugar, right? Phosphoric acid attached to 5th carbon of pentose sugar. So what is the correct answer? The right answer of this one is option A. Am I clear? Yes. Last question, beta. Today's last question. Very good. Last question also, I hope you will give very easy answer. Very easy. All of it, try fast. Next class also we are discussing most important biotechnology questions. I hope this session you like, and this important question try to recall once again where I am doing mistake, which topic I am very weak, just to try to recall all of you. Which of the following are all nucleotides? All nucleotides. Come on, answer. Come on, answer. Sir, my birthday, I'm coming soon, please. Okay, my dear, don't worry. Happy birthday. I'll give you a nice gift. Come and meet me. What is the correct answer? All nucleotides asking. Adenosine, nucleodylic acid, cytidyllic acid and cytosine. Adenylic acid, cytidyllic acid, guanylylic acid. Cytidine, cytidine, adenine, adenylic acid, uracil, thymidine, thymidylic acid. Very simple answer. Very simple. Yes, wherever all acids are there, phosphoric acid attached nucleotide. So I am going with option B. Shall we have one more question? Will you stay here? Shall we have one more question? Yes. Yes. Shall we have one more last question? Are you ready? One question. Don't leave. Last question. Can you? Last question. What is the correct answer of this one? Accession and reason. Something given better here in accession. In a process of transcription, template strand with polarity 3 to 5 play a major role. Correct or not? Just tell me. The sir statement. Accession is true or false? Just tell me. Accession true or false? Just I want. In a process of transcription, template strand with polarity 3 to 5 play a major role. Correct or not? Just correct or not? True. This statement is true. Yes, it's true. 5 to 3 is a template strand. Correct. So 3 to 5 is a template strand. Correct. Reason. DNA dependent RNA polymerase. I told you this is very cute one enzyme. DNA dependent RNA polymerase catalyze the polymerization only one direction, that is 5 to 3 prime. Is it correct? His statement is correct. Come on, come on, come on. His statement is correct. Come on, come on, come on. Is this statement is true or false? True. Yes, true. The statement is true. It will be from 5. This is 3 dash to 5 dash of template strand, my dear. So RNA will be formed from where? RNA will be formed from 5 dash to 3 dash. That's why I told you RNA always from where to where? 5 to 3 dash. Statement correct. So the right answer is what? Come on, come on, come on. Right answer is what? Option A. This is a right statement. Both statements are correct and this is the right. So I can go with what? Option A is the right answer. You got my point? A is the right answer. That's why I'm telling you, but my dear student, this session exclusively for you all. Brilliant is conducting 16 days. Don't worry, no problem. Small mistake you will do before final examination. Try to correct, try to rectify. Oh my God, this chapter I'm very weak. Open that chapter and revise. Whatever the question we are discussing, try to attend and learn something. Get 100 out of 100 marks in all subjects. You got my point? Right. Okay. Thank you very much for attending this session. Thank you so much. Next class, biotechnology application. Both chapters I am going to discuss. I hope you will come and attend, and I hope you enjoy this session. All the very best for the final exam, student. Thank you.